And cyanobacteria actually, they have been used for quite a long time in basic biology as model organisms, especially in research of photosynthesis, its mechanisms, and its acclimation to different environmental conditions. And what are cyanobacteria? I suppose many of you are not. The model organism we are using is Synecocystis 6803, and we are using it because the photosynthesis of this organism resembles that of the organism. So we think we can apply our results to higher plants, and this is much easier in many senses to use in the laboratory compared to higher plants. But generally, cyanobacteria are prokaryotes, and they are unique prokaryotes since most prokaryotes have a so-called outer membrane and plasma membrane. But these cyanobacteria contain also thylakoid membranes like plants. And these thylakoid membranes are the site of photosynthetic light reactions. Cyanobacteria have contributed to the evolution in a very special and important, decisive way. Namely, they were the first organisms on the Earth to produce bulk amounts of molecular oxygen through oxygenic photosynthesis. So that means that they are responsible of early oxygen formation in the atmosphere and the conversion of the reducing atmosphere to an oxidizing one. And they are also so responsible of the rich evolution that has resulted from the oxygen-rich atmosphere. Furthermore, plant chloroplasts stem from ancestral cyanobacteria, according to the endosymbiosis theory. So cyanobacterial cells have invaded other cells, and they have been the starting point for plant chloroplasts that are the site of photosynthesis in plants. Now, oxygenic photosynthesis performed by plants and cyanobacteria is the main biological process on Earth where the light energy from the sun is converted into chemical energy. And all higher life on Earth receives its energy directly or indirectly from photosynthesis, and all oxygen in the atmosphere is evolved during this process. Cyanobacteria were earlier called blue-green algae, but they shouldn't be called algae because they are prokaryotic cells, and algae are eukaryotic organisms having completely or very much different cell structure. Where do cyanobacteria live? In all kinds of waters and also terrestrial environments. So very varied environments, varying, for example, according to the temperature, very much. And this Synecocystis, what we are using, has been isolated in the 70s from fresh water. Many species are also symbiotic. Some species can fix atmospheric nitrogen. Some strains produce toxins, as you have read in the newspapers, during these cyanobacterial blooms, especially in polluted waters during the late summer. They are really very harmful. And also they live in many scientific laboratories around the world, and they are actually used actively in research. I will try to describe you more closely now why they are so useful for basic and maybe also for applied research. Synecocystis 6803, our beloved cyanobacterium. Its complete genome structure was determined in 1996 as the first one of all cyanobacteria. Already then it was used in photosynthesis research, but this formed the important basics for modern systems biology research. It has a 3.6 megabase long genome with 3,168 putative protein coding genes identified by similarity searches and computer predictions. Of the putative genes, about 45% are assigned with known biological functions. And of these, 139 are needed for photosynthesis and cell respiration. About function of 55% of the genes, there is no knowledge currently, and also that is also one reason why this organism is important, because it gives good opportunities for us to clarify the significance and cellular role of these so far unknown genes. So the information currently in Cyanobase allows a variety of experimental methods to be utilized for elucidation of function of each gene, and also for unraveling of regulatory networks when controlling gene expression, like systematic gene disruption project, transcriptome and proteome technologies at the genome-wide level. So this transcriptome technology means that it's possible to study function of practically all genes, expression of all genes, when we have an organism where the whole genome sequence is known. And this proteome technology aims at identification, localization of all gene products. But that has much more restrictions compared to the transcriptome study. But anyways, nowadays about almost 10% of Synecocystis proteins have been identified and localized in different parts of the cell. So the cell of Synecocystis looks like this. It's actually a typical bacterial cell, containing first cell wall, outer membrane, peptidoglycan layer, plasma membrane, this typical bacterial cell wall structure. Then there are these thylakoid membranes that fill in most of the cell, and everything that belongs to cell structures, ribosomes. And these black spots here are the light harvesting machinery that these cells use to absorb light energy. So that was a schematic presentation, and this is an electron microscopical view to show you a more real image, and also to demonstrate that the cell structure is affected by environmental condition. In this case, these cells have grown what we call autotrophic cells. They have grown on light energy, so they need only mineral nutrients in addition to light. These have grown on glucose with no light, so they are not growing on photosynthesis, and you can see that these thylakoid membrane layers are mostly disorganized, and the cell structure has really changed quite a lot. Okay, an example of a proteomic study. So we can isolate proteins from different parts of the cell, and then we run them on two-dimensional gels. First we separate the proteins according to their PI, and then according the next dimension according to the molecular weight. And here, when we use cells that have been grown under different conditions, first, we can identify all these protein spots via mass spectrometry. But here we can compare what proteins are important under every conditions, because we can see large changes. For example, this area contains a lot of photosynthetic proteins that are important during autotrophic growth, and here the area looks completely different when the cells don't photosynthesize. And we can see several new spots that have been arising when the cell metabolism has been changed. So that was cyanobacteria and Synecocystis in very short. So what is then photosynthesis? Some details about it. Unfortunately, this is not very strong. I hope you can see. But anyways, I say briefly. So photosynthesis consists of light reactions and dark reactions. And the aim of all is to convert light energy into chemical energy that is stored in form of sugars. The site of light reactions are the thylakoid membranes inside the cell. The site of the dark reactions, enzymatic reactions, is the water-soluble part of the cell or of the chloroplast. And the aim of the light reaction is to produce ATP and NADPH. ATP is used as cellular energy, and NADPH is used as reducing power in the dark reactions. So let's have a look how ATP and NADPH are formed. First, every light quantum is exciting a reaction center chlorophyll molecule in photosystem 2 or in photosystem 1. And then an electron transfer chain starts to carry the electrons through NADP plus reductase. And here the electrons are drawn from water. So in the very beginning of photosynthesis, after light energy has been absorbed, water is split and oxygen is released. Okay, and then chemiosmotic ATP is formed chemiosmotically because at the same time with its electron transfer, a proton gradient is formed across the thylakoid membrane. The current model of the thylakoid membrane says that these components, enzymes redox components that carry the electrons, are embedded in the thylakoid membranes, and the proton gradient is formed by several reactions. First, splitting of water produces hydrogen. Then, next, an electron transfer step pumps protons across the membrane, and also when NADP plus is reduced at the end of the light reactions, so protons are bound into this structure. So these all together form the proton gradient across the membrane that is utilized in the chemiosmotic synthesis of ATP. And these NADPH and ATP are utilized in the enzymatic reactions for binding carbon dioxide into sugars. So, then the short part of my talk, considering if cyanobacteria have potential as life-supporting oxygen producers under extraterrestrial conditions, when they can grow in so many different environmental conditions, and they produce oxygen efficiently, so can this maybe be utilized in under even more environmental conditions. But to answer this question, cyanobacteria shall be characterized under specific conditions. Currently, we cannot say. But what should be done is to analyze capacity to grow and produce oxygen photosynthetically, perform studies at the transcriptome, proteome, metabolome level. And with my colleagues at Turku University, we have just started to consider this idea, and we have started to test Synecocystis under low atmospheric pressure. And this is the experimental setup what we use. We have the cell cultivation, the liquid cultivations in a growth chamber, where we can adjust atmospheric pressure and temperature, for example. We will control also other parameters, but now we go further step by step, and we are at the very beginning now. But we have gone down to 0.25 atmospheric pressure. And what we have seen there has been quite interesting and preliminarily promising. Namely, here I present now the result at 0.5 atmospheres, but we get exactly the same when going to 0.25. Next week we will go to 0.1 in the atmospheric pressure. This is the control cultivation going normally as bacteria do, and we take a sample every day and monitor the growth in the liquid culture. And these 3 replicates have grown under low atmospheric pressure. And as you can see, they don't grow much, but if they are after 10 days put to normal atmospheric pressure, so they start to grow completely normally. So they are not harmed in any way. They are not suffering so that they couldn't recover after the stress is removed. But do they produce oxygen here is an important question when they don't grow. And the answer is yes. Actually, what we have done is we have taken samples at day 4 and at day 10 and measured oxygen production immediately, the photosynthesis oxygen production. And actually we have measured also whole photosynthesis. And they take place completely normally. Even if the cells don't grow, they produce oxygen completely normally, what is really important. So, this new observation that Synecocystis is able to produce oxygen normally via photosynthesis in spite of lack of growth under low atmospheric pressure leads to the next question. Is it possible to find conditions where it will also grow and therefore produce even more oxygen? And that's what we will focus on now. If we can add something into the growth conditions so that they start to grow. But so far we have done only these very first growth characterization experiments, and I'm considering these ideas together with my colleagues Eera Kanervo and Kirsi Lehto at the University of Turku Department of Biology. So thank you for your attention.
Thank you very much. The paper is open for questions.
You said that the cyanobacteria are the bulk producers of oxygen in the first place.
Yeah.
When does that take place?
Well, the atmosphere started to change significantly from the reducing one to the oxidizing one 2.7 billion years ago. But already a long time before that, the cyanobacteria, of course, had evolved and started the oxygen production.
You need first to oxidize the rocks and the minerals, iron, before the oxygen goes in the atmosphere. That's why you have already long time biological oxygen production before it releases in the atmosphere. That's completely correct.
Yeah, certainly.
Yeah.
So now, I would like to discuss here a little bit this topic, which is kind of like maybe consequently related to these experiments. Now, if we are testing cyanobacteria as an organism, photosynthetic organism is producing, and now maybe as a target, whether it can be used as a life support conditioning on Mars, for instance. So, nicely, possibly, we can photosynthesis can produce oxygen even under low atmospheric pressure, then maybe in culture with some contained greenhouses where the radiation from the cultures will survive there. Maybe even if they do not replicate very much, I think that's just kind of sitting there and photosynthesizing.
Yeah, then only the culture has to be strong when put there.
Yes.
Yeah.
But the question, of course, then is that whether it would be safe ever to establish this type of organic photosynthetic cultures on Mars. And realizing that these organisms actually may survive also in terms of contamination of the planet. And at least you are familiar with this contamination risk. So what you say, can please be taken there?
I think there are two things which we have to separate, two possibilities where cyanobacteria might be interested. One is as a biological system in a life support system, in a bioregenerative life support system for the astronauts. And that will be in a cabin and will be in a closed environment. And the other one is that there are really questions whether something like cyanobacteria could survive on the surface of Mars under certain conditions. And you have then to simulate the environmental conditions. Is that okay? Let's come first to the life support system. There is what is required that you need, because you cannot bring all the oxygen and all the water and all the food with the astronauts if they go for 3 years to Mars. The whole trip will take about 3 years. On the space station, most of the life support systems are brought up and done. There's only a oxygen recycling, more physical-chemical. And everything else is just released in space, or brought back to the Earth, or burned in many places. So I think cyanobacteria can be used for oxygen production, so recycling the air, using the CO2 and forming the oxygen there are some calculations that they are very efficient in that. They can also be used as food. I mean, nobody wants to eat cyanobacteria, but if you make a nice pudding or stir, they can be used. The only problem is I think they have too much phosphorus or something. So they cannot be 100%, because single cells are not such a good diet for people. And there is a study which is called MELISSA. Don't ask me what the acronym is. A European study which is already very far advanced, where they have the different compartments with bacteria and so on, and one is the plant and algae, but I think they take cyanobacteria compartment. And I can give you the reference for that. This group has already done a lot of studies, and when you want to do these things, you should first try to look what they have done. I think this is the only group which I know have done studies with cyanobacteria. I think they have used cyanobacteria. All the others have used either higher plants, I mean there's an American study or Russian study, but they mainly use higher plants. So actually we have also a diploma by evaluating the different systems, bioregenerative systems, and this person, Mark Chupada, he also had a presentation at the last IF conference and so stuff. We found out that cyanobacteria are the most efficient in air recycling. Not for food, but in air recycling they are the winners. So in that case it's interesting to work on them. But then you need them to keep them just in a closed environment, and you have somehow a continuous culture or insect culture or something, cellular culture.
Maybe I'll just say one thing that may be important. That cyanobacteria, like the scientists, they can maybe be used also as cell factories to produce important compounds. So in addition to oxygen.
Yeah. But then,
Chemically modified cells.
but then you need what kind of compounds? Do you think vitamins or something?
Yeah, organic compounds.
Yeah, but then you need some factory behind them to extract it or something.
Well, but if they are eaten as such,
Yeah.
or as a pudding.
Yeah. Yeah. I agree completely that part of it can be eaten, but they produce more than you can eat. Because you cannot live 100% from
No.
And the second one is, if I understand your experiments right with the reduced pressure, but they are still in a aqueous culture. So what happens if you have a lower pressure in the air? What happens with the aqueous culture? Is that somehow improved? I mean, there will be some evaporation.
Yes. The total air pressure is measured by the air pressure adjustment.
But they live submerged in the water.
Yes, and we now have the temperature and vaporizing phase is not reached yet, which is kind of convenient because like cyanobacteria requires 30.
About 30 degrees.
Degrees of temperature, so at least at this pressure yet, the water is not very much.
Yeah, I'm just thinking, if you want to do these studies, I think the aim is to see whether they can survive in some areas or even grow in some areas outside on Mars, not in the habitat, but somewhere outside. Why don't you look for take such cyanobacteria which grow on rocks, in rocks, so which don't need to swim in water, and to be submerged in water?
Yeah, we have considered also maybe actually to try other species, but we first want to make this basic characterization with this organism that is known so well in many senses. But these can also grow on kind of solid. This can grow on agar. So it's not the only way to calculate this. But this is the way to get the most biomass.
You had a comment,
Question actually about the energy production cycle. Is it only based on photosynthesis, or is there a cycle feedback loop of ATP as in higher species?
Yeah, there is also cell respiration taking place. Yeah, but the question how much that is actually needed in the light, I think it's not very efficient. But in the light they can produce ATP, they can produce reducing power based on photosynthesis, and because these are prokaryotic cells, so everything takes place more or less in the same cell compartment. So it's not like in eukaryotic organisms that the cell is divided into several compartments and photosynthesis takes place in one and cell respiration in the other, and so this is the most simple system.
I had a question regarding numbers. Could you maybe give us some numbers about how much oxygen is being converted?
Yes. So we express it per chlorophyll and per time. So we measure how much chlorophyll these cells contain.
Yeah.
And they produce about, it varies according to the age of the culture. The younger cells produce higher amounts than old cells. So about between 1 and 500 micromoles of oxygen per milligram chlorophyll per an hour. Varying depending on the age of the culture.
So between 100 and 500 micromole
oxygen produced per milligram chlorophyll per an hour.
Right. And how does that compare then to plants, for example?
It's comparable to plants. Yeah.
I want to know whether do you have any plans for an experiment with polarized light, the circularly polarized light, etc. Because there is a suggestion that the circular polarization, polarized light can lead to chirality changes. So
yeah, that might be one thing we have considered different kinds of stresses to be studied one after the other. Like UV light is one, different gas combinations, and polarized light is one, as you said. Indeed. But we are now at the very beginning. So next we try to get the cells to grow, if possible.
I mean, this is of course a remarkable suggestion. I mean, so he would suggest to expose it to polarized light of the wrong sense, such that you would actually convert your cyanobacteria into a new cyanobacteria of a different habitus. I mean, this is not just some small kind of stress. It's a complete revolution, of course.
Like we can give radiation and cause mutations in the cells.
Yes.
And that has been the traditional way to search for better capacities to grow under certain conditions.
So, yes.
How is the reaction to UV? I mean, in the old days it could function under UV conditions. Do they retain that capability?
That's what we haven't tested so far, so I cannot say, but it's one of our experimental conditions what we have planned to include. I suppose they survive. Of course it depends on the efficiency of the UV, but they certainly tolerate it to some degree. But UV has the tendency to cause mutations, and it's also deleterious to photosynthesis and so on. So we don't know how efficiently they, for example, produce oxygen under UV light. But that's what we want to test.
I mean, in addition to that, I think they have developed a special UV screening device, so they can somehow screen, also use that to screen the harmful UV radiation. I don't know whether that's
it's certainly sufficient to a certain amount of UV, but what is that amount we cannot say, and it has not been studied in cyanobacteria.
We had the same kind of problem with our films. We wanted to enhance the mechanical stability through polymerization. So we used some polymerizable lipids and was successful with this. But simultaneously the UV radiation destroyed the biofunctional lipid. So it was of no use. And this was different from different one to another. I mean, depending on the UV, there is several types of UV. So it will be interesting to hear what you find out in the future.
What will happen if you remove the UV source Does it go back to this repairing automatically?
Since once questioning, it remains.
There is no repairing mechanism available.
Well, we did not maybe study that long cardiac study. We just told us that it was not good for us.
Oh, okay.
We forget all about the problem, but it was not good for us.
No, the reason why I'm asking is that because there's only very efficient can survive the kind of thing, and for which, of course, we have enough time at our disposal. That's not only we have enough time.
It's always coming UV from place.
Yeah.
It's just a matter of time.
Yeah.
When it's enough.
That's
Normally you get when you absorb the energy just in excited state and then it results in covalent bonding, and this covalent bonding is irreversible. Some cellular mechanism starts to repair. But chemically, it's most UV products are irreversible. Some are reproduced thermal, they buy, most of them cannot be reversed.
So I would like to ask still once more, although you're a little bit distracted already, so you persons who are kind of maybe involved in or at least participating in the discussion of the Maenpaa mission, so what exactly is planned about the life support facilities?
It's more general discussions.
There's nothing planned so far. There are some individual groups who do, and I must say the biorational life support development is at the moment not very much supported. It could come again now with all the ideas of using missions to Moon and Mars. In the States, it's nearly to zero. There's not very much going on at the moment. There was a lot of work done at NASA Ames, at the JSC, but it's at KSC also, but there's not very much at the moment. Probably it will come up again. In Europe, they support the ELISA system, which is quite interesting, but it's more on microorganisms and up to algae and cyanobacteria. So it's probably more a model system. And they have already tested it with rats. So rats can survive just in this kind of cycle which produces everything they need. And in Russia was a very efficient what they call BIOS-3. There they had humans for a couple of months in a closed environment, it's about subsurface, so everything was closed. I think the only thing what was added was some animal food. They had lived only on plants and added some animal food. But that is also not very much healthy because of lack of financial support. So I think if one really wants to send in 20 or 30 years humans to Mars and even earlier to the Moon, it's something which needs to be developed. And I know that ESA in the European Space Agency in the Aurora program, if your country joins it, they will probably spend some money in development. I will anyhow spend some money, and if your country joins the Aurora program, you can participate. And the Aurora program that ESA has is a program which includes the developmental steps, including technology development, but also robotic missions towards human missions to Mars. That is the final goal of mission. But I mean, the next 20 years will be all the preparatory steps in that direction. And it's an optional program that countries can join or not.
Still have plenty of time for discussions.
I'd like to follow up my question about this Martian thing. See, the thing which you are saying about if you are going to take the cyanobacteria or whatever chain is there, are we not going to pollute the atmosphere already, and whether the Martian things will change, because you are saying about so many other experiments also planning, whether does it change the Martian environment irreversibly, or whether there is there any study about those things?
I'm just joining a team at the moment who discusses biohazards and planetary protection with regards to human missions to Mars. So it's something one is certainly aware and is trying to find out. So it's a very important point you have raised. There are no solutions at the moment. There are guidelines for protecting the environment and also not bringing microorganisms or any living thing to a planet until you know whether there is some indigenous life or not. For robotic missions, there are very well developed guidelines, and they are adopted by COSPA, the Committee of Space Research, and all spacefaring nations, even laid down by the Treaty of the United Nations, have to follow this. It's all for robotic missions, it's all exactly laid down how much you have to sterilize depending on where you go and what planet and what region of the planet. But for human missions, it's at the moment open. So these are things one has to discuss and think about, because it's inevitable that when you bring humans to a planet, you will also bring the environment of the humans, including microorganisms, including, if you don't have a completely closed system, including gas and other waste products which will go into the mine. It's a very critical point, and one has to discuss it. We have even, with a colleague, now published a paper in Space Politics to think early of maintaining some what we call planetary parks, like national parks, to keep some areas completely free of human interference. I think it's something one has to think about. But of course, the air will be globally.
I find it interesting that you mentioned now, I think several times, that it has to be first determined whether the planet has indigenous life. Does this mean that if it does not have indigenous life, then it will not be so catastrophic economically? Like, is this a crucial question of, well, if it's barren and we can do it? Or is it still as bad?
I mean, what's bad and what's good? Planetary ethics, I think that's something also space law and space ethics have to find out, like an Arctic treaty or something one has to have. There is a moon treaty, but not all countries have signed it, except the United States, including the United States, that have signed it. The moon treaty to treat the moon similar like Antarctica. Nothing belongs to any country, and one has to somehow take care of it to lose it. And probably there will be something like that if we go more to Mars, similar thing. I mean, what happens if we find life there? It will have a big impact on all the future of research. In what way, I don't know. Whether we will especially go there and have a look, or whether we say we can send humans and keep it with Martian life, don't pollute it, I don't know. It's not solved, but has to be solved. I think this needs to be learned with people doing ethics, politicians, not only cannot answer by science. Yeah.
If you were like the long run, keep that kind of way. Like,
You think we will?
we will go there eventually.
We will.
Yeah.
Yeah.
In the long run.
Yeah, I think we will go there.
No stopping us.
Yeah. It's essential, I believe. So the young people will witness it.
Because such a prediction was expressed at the time of lunar missions also, and lunar missions really did take place.
Lunar missions for sure, yeah. But lunar missions are not such a problem.
Yeah, I agree.
But they didn't know that then. I mean, they didn't really know until they came back.
Yeah, they were kept in quarantine, so.
There's no life, it's ours.
No, but I mean, what I already told Kelsey, that if you want to do studies on such single cell organisms or whatsoever under simulated Martian conditions, that there exists a tropical team established by ESA on it's called responses of organisms to Martian environment. And the tropical team means that a group of people have submitted a proposal to ESA just to have a pool, a discussion group where finally proposals can come out for something. And so I can give you the link or even my email address is sufficient. And they meet, I think the first one is now finished at the end of this year, but probably it's healthy and productive and can even continue. And there are different European groups who have either Mars simulation facilities or have some organisms like pyrophilic organisms or also people working with lichens and we are with our spores again. And to see how we can jointly then propose experiments which we can do using one or the other of these facilities. So I think that's for everybody who's interested in this open team and one can join this, especially for the Europeans.
Regarding this earlier question which Roman Rai had about the circular polarized light, only to perhaps rephrase it, unless I did misunderstood the question already. Is there any knowledge about the consumption or the conversion of air to high oxygen as a function of the polarized light? If it's for right-handed or left-handed polarized light, is the efficiency or is of cyanobacteria different? Is that known?
I don't think so.
No?
She's saying that she's just starting the study, so polarized light, no polarization has been seen. I mean, they are not used polarized light at all.
No.
So no study.
It's a really such a straightforward thing to try, right? Even better. So
that's why I was surprised that, especially when it has so much implication for chirality.
Yes.
Such studies would be.
So how long will it take you to do such a thing?
Well, there are a few steps now we have to do. First actually to see if they can grow, if it is something, maybe something that they grow, that's the question. But it also depends how efficiently we can continue this experiment. So now we have proceeded only one step after steps.
Yeah.
Plan to keep them very far.
I mean, how would you?
It would still be interesting just to look at polarized light even on the standard conditions. I mean,
Yeah, so, yeah,
so it would be done very easy.
Yes.
So, I mean, do you even have polarized light in your lab? Or do you need
No,
somebody?
we don't have. No.
But when somebody
It's
told
purpose
you,
and physicists.
yes, I'm just concerned that I mean, if it has been forgotten for the last 20 years, I'm just concerned that it may be forgotten for the next 2 months already. So I'm very curious to know what the result is. Very curious.
No, what exactly is the thing which prevents you from? You said that we have to see whether we can proceed for the next step or something like that. What is the thing which prevents you from having a polarized light and then doing that thing? Is it a very too difficult kind of a step to?
No, I don't think so. No. When you get one that's
Polarized light
it's
is
actually
actually
quite
quite
easy. Very easy.
I
I'm sure that water slide and the condition is something which is quite often done.
Although I'm quite skeptical whether the polarization of light would affect the photosynthetic processes. It is just capturing the exciting energy of the light one.
No, but actually there are some computations have been done which show that there is definitely change between the left and right hand side because of this particular thing. They have the calculations and they have done it with the problem was whether we can totally separate for this light or something like that from the light. We are talking about some resources, but laboratory sources of pure purpose light is not a difficult thing to have. Right? So that being the case, one can see the thing is we have there is a lot of debate going on about the polarized light coming, whether it's supernova that produces separate polarized light or not is something that is a different debate. But they already showed, I think theoretical calculations have been done, and then they show that it definitely leads to chiral change.
In
amino acids, the sugars, essential sugars.
In chemical reaction. But photosynthesis is an energy capture reaction, so. But this will be tested for cells.
The efficiency can be different from our science.
So actually the thing is that the calculations have been done for the astronomy. So that definitely stability wise of the molecular wise, we have quite sugar is mostly separate resources. These are all calculations that have been done. That's the reason why I thought that it would be interesting to have an experimental verification of that when you have got the setup already. Yes, we can have that.
So just to give us a picture, how do you actually measure the production? You just have the bacteria in a container. They grow
We
into
do
starch.
it in a so-called Clark-type oxygen electrode. So there is an electrode that reacts to the amount of oxygen in the medium. And so we put the cells there in the growth medium. And then we put light on.
Yeah.
And we see increase in oxygen concentration. And when we switch light off, the oxygen production is finished. So we know it's photosynthesis.
Mm.
And then we calculate how much oxygen produced. This is a standard protocol that is used a lot in plant research. So
Wow.
we can use whole cells in this system.
Yeah. So the whole procedure is fairly accurate.
Yeah.
Very impressive. Very exciting.
I'd like to mention something about cooperative in this system. I think this is a way to provide. Right. If no more, maybe we can go further. I give the word to the next chairperson, Chris Fridge. And let us once more thank the speaker also.